things we'll speak about here as it applies to neural integration: summation, which we kind of touched on already, the idea of adding several graded potentials together. If we think about adding an IPSP to an EPSP, both of those cancel out. If we think about adding an EPSP to an EPSP, that's a stronger depolarization that would get us to threshold. If we think about adding an IPSP to an IPSP, that's going to take us even further away from our threshold. All right. We'll speak about frequency coding, divergence and convergence, and then the axon, hillock, and threshold. Now, as far as neural integration goes, this is basically looking specifically at the axon hillock. We talked about the axon hillock before. It's that really critical region on the neuron where we decide, do we have enough of a membrane potential change that meets that threshold requirement, okay? And so neural integration is a way for us to kind of analyze all of the different types of signals that are coming in to the cell body, to the soma of that neuron, and then determining at the axon hillock, does this meet the requirement to fire an action potential? Do we get to threshold, okay? If the potentials are below threshold, there will be no action potential. If the potential is at or above threshold, there will be the initiation of the action potential, okay? And so the first things that we want to think about as it relates to neural integration is that we have different types of transmission of these signals on different relations, on different interactions with different neurons. So we can think about divergence as the splitting of a signal. So we have a presynaptic neuron synapsing onto several different neurons here. Okay, so if I'm releasing some neurotransmitter here, this signal is going to be divided among 4 different neurons. That's basically going to water down my signal, okay? And that will influence whether or not this signal is strong enough to initiate an action potential on each of these adjacent neurons, okay? So that is how divergence plays a part in neural integration. On the other hand, convergence also plays a part in neural integration as well. If I have many different neurons synapsing onto one soma or one cell body, then I have several different graded potentials, coming onto my neuron, and then all of those signals will be analyzed, and they could definitely get us closer to threshold. Okay, they're going to basically amplify the signals that are coming in because there are so many neurons synapsing or converging onto the postsynaptic neuron. So divergence kind of splits or waters down our signal. Convergence kind of amplifies or aggregates our several different signals coming in from several different neurons. All right. The other thing we want to talk about is summation. And we introduced the idea of temporal and spatial summation. So this is kind of just review at this point, or reiterating that idea. We can basically add different weighted potentials if they are close enough in time. That is called temporal summation. So it's happening at the exact same spot, okay? So we think about site A here. And if I have one graded potential depolarizing the membrane, if a very short amount of time has elapsed and then I have another graded potential also depolarizing the membrane potential here, I can simply combine those 2 graded potentials. And as we can see, because they were both EPSPs, they actually meet that threshold requirement, and then I can fire an action potential, okay? So that is temporal summation. Spatial summation, on the other hand, is if I combine several different signals that are happening at different spots, but they're happening at the exact same time. So I am going to combine the graded potential happening at site A, which is an EPSP. I'm going to add that to the graded potential happening at site B, which is also an EPSP. And then I'm going to add that to the IPSP, which is happening at site B, because they are happening at the exact same time, but at different spots on the neuron. So I have A, which is a depolarization, B, which is a depolarization, C, which is a hyperpolarization, And as we can see, that actually gets us closer. If we add A and B, right, two depolarizing events, that gets us closer to our threshold stimulus, and then we can fire an action potential. If I were to add site A and C, so the greater potentials coming in on synapse A and synapse C, because one is depolarizing and one is hyperpolarizing, those would completely cancel each other out and there would be no membrane potential change, okay? And so this is how summation also plays a role in neural integration.